In 1895, German physicist Wilhelm Conrad Röntgen discovered what are known as X-rays, also called Röntgen rays, which were named after him. This revolutionized medicine. Suddenly, it became possible to take a look inside the human body without having to use a scalpel. These days, not just X-rays, but also many other imaging methods such as CT, MRI or endoscopy are popular diagnostic and treatment methods respectively. Molecular imaging counts among the most modern imaging methods. With the help of illuminated particles, diseased tissue is illustrated at the molecular level.
The new modalities which we develop here are called molecular imaging technologies. And the reason is that we don't look at structures, we look at molecular functions. We look at single molecules even in the function of the organs. So now these modalities will add very new information about molecular pathways involved in pathologies of the heart and the vessels, and not only look at structural levels.
Professor Michael Schäfers is the spokesperson for the Collaborative Research Center 656 Molecular Cardiovascular Imaging at the University of Munster. Imaging methods that are meant to be used for diagnostics and treatment monitoring for heart diseases are being developed here.
The problem with the cardiovascular diseases, on one hand, we have a lot of incidents, so there is a high incidence for cardiovascular diseases. On the other hand, the body is intransparent, so it's hard to look at the heart, really, and look into the body. So this is why we need new imaging modalities.
To do this, the scientists need so-called tracers that make the diseased tissue visible.
First of all, we have to define what we call targets, molecular targets, which are molecules which are regulated in the disease process, such as in cardiovascular diseases. Now, what we need to develop, and this is why chemistry is involved in the Collaborative Research Center, is drugs that we can send in, and these drugs are targeted against the target. That's why they're called tracers. So they seek their way, they find their way to the targets, accumulate at the target site, and then, because they are labeled by, say, radioactivity or optical fluorochrome, so fluorescent parts, they send out the signals to the outside of the body, and we measure from outside these tracers.
To illustrate the accumulated data in images, biologists and chemists now need the help of their colleagues. Now it's the turn of the physicists, mathematicians and information scientists at the CRC 656.
So one of our aims in our research is that we would like to improve medical imaging. And one of the major problems in imaging is that you may have some motion that leads to blurring in your images at the end. And for example, in techniques like PET, positron emission tomography, which takes several minutes for the acquisition, the heart is beating due to its own contraction and also due to breathing. So you have two different types of motion that leads to some image blurring at the end. And this is where we would like to develop new methods to detect this kind of motions and also to correct for this motion.
So our approach is actually to consider the different motion stages, separate them, try to establish the motion from one stage to another, and given that information, we can correct motion, we can remove the motion. So after this removal, we can put all the motion corrected data together, and this way we can produce high quality image data without motion, and this should be useful for physicians for quantification and diagnostic.
So one issue, of course, is then at the end to prove that your methods work correctly. So one way is, for example, that we build up a hardware phantom like Wilhelm, which produces some data that we can use to simulate motion, and which also produces some ground truth data where we have no motion at all. And we place this phantom inside imaging technology, inside a tomograph, and generate then some simulation data which we can then use to prove our methods.
So actually we want to validate our algorithms in multiple ways. One way is already mentioned hardware phantom, and there are two different ways, other different ways, and one is using mathematical models. We can obtain mathematical models of body organs from real measurement data, and then simulate data for our algorithm validation. And also, we are using real patient and mouse data for validation.
Before the methods can be used on patients, the scientists test them on animals.
So that's what we call the translational part, and it involves animal experiments. And these animal experiments are very focused on using a few animals and applying the same approach that we would do in the future in patients. And in these mice, we try to see what we would like to see in patients in the future. So we inject the same probe. We have very dedicated imaging modalities which have a very high resolution. So this is a very easy and translational step. So we test it in mice first, and if it is successful there, we can kind of straight away go into humans and see if it works there. In our collaborative research center, we now work for, say, 8 years on the topic of cardiovascular molecular imaging, and we are very proud and looking forward to applying these methodologies to patients. So in the last weeks, we've done the first studies in patients, and we see signals that we also saw in mice. So we are quite confident that we can translate our methodologies from mice into patients.